The principal glands of salivation are parotid, submandibular, and sublingual glands. In addition, there are many tiny buccal glands also. Daily secretion of saliva normally ranges between 800 to 1500 ml with an average secretion of 1000 ml per day. Saliva contains two major types of protein secretions, serous and mucus. Serous secretion that contains ptyalin, which is an enzyme for digesting starch. Mucus secretion that contains mucin that helps in lubrication, and it also has surface protective purpose. The parotid gland secretes almost entirely the serous type of secretion, whereas submandibular and sublingual glands secrete both serous secretion and mucus. The buccal glands secrete only mucus. Saliva has pH between 6 and 7, which is favorable range for digestive action of ptyalin. Saliva contains specially large quantities of potassium and bicarbonate ion. Interestingly, concentration of both sodium and chloride are several times less in saliva than in the plasma. Salivary secretion is a two-stage operation. First stage involves the ACINI and the second stage involves the salivary ducts. The ACINI secrete a primary secretion that contains ptyalin with or without mucin in a solution of ions with concentration not greatly different from those of typically extracellular fluid. Firstly, sodium ions are actively reabsorbed from all the salivary ducts, and potassium ions are actively secreted in exchange for the sodium. Therefore, the sodium ion concentration in the saliva becomes greatly reduced, whereas potassium ion concentration becomes increased. However, there is excess sodium reabsorption compared with potassium secretion, which creates electrical negativity of about -70 mV in the salivary ducts. This negativity in turn causes chloride ion to be reabsorbed passively. Therefore, the chloride ion concentration in the salivary fluid falls to a very low level, matching the ductal decrease in sodium ion concentration. The bicarbonate ions are secreted by the ductal epithelium into the lumen of the duct. This secretion is least partly caused by passive exchange of the bicarbonate for the chloride ions, but it may also result partly from an active secretory process. The net result of this transport process is that under resting condition, the concentration of sodium and chloride ions in the saliva is only about 15 milliequivalent per liter each. That is about 1/7 to 1/10 of their concentration in the plasma. However, the concentration of the potassium ion is about 30 milliequivalent per liter, that is 6 to 7 times as great as in the plasma, and the concentration of the bicarbonate ion is 50 to 70 milliequivalent per liter, that is 2 to 3 times that of the plasma. During maximum salivation, the salivary ionic concentration changes considerably because the rate of formation of the primary secretion by the ACINI can increases as much as 20-fold. This acinar secretion then flows through the duct so rapidly that the ductal reconditioning of the secretion is considerably reduced. Therefore, when copious quantities of saliva are being secreted, the sodium chloride concentration is about 1/2 or 2/3 that of plasma, and the potassium concentration rises to only 4 times that of the plasma. The salivary glands are controlled mainly by parasympathetic nervous signals, all the way from superior and inferior salivatory nucleus in the brainstem. The salivatory nucleus are located approximately at the junction of medulla and pons, and are excited by both taste and tactile stimuli from the tongue and the other areas of the mouth and pharynx. Many taste stimuli, especially the sour taste caused by acid, elicit copious secretion of saliva, often 8 to 20 times that of basal rate of secretion. Also, certain tactile stimuli such as the presence of smooth object in the mouth causes marked salivation, whereas rough object causes less salivation or occasionally even inhibits salivation. Salivation can also be stimulated or inhibited by nervous signals arriving in the salivatory nuclei from higher centers of the central nervous system. When a person smells or eats favorite foods, salivation is greatly increased than when the disliked food is being smelled or eaten. The appetite area of the brain, which partially regulates this effect, is located in proximity to the parasympathetic center of the anterior hypothalamus, and it functions to a great extent in response to signal from taste and the smell areas of the cerebral cortex or amygdala. Salivation also occurs in response to reflexes originating in the stomach or the upper small intestine, particularly when irritating food are swallowed or when a person is nauseated because of some gastrointestinal abnormality. The saliva, when swallowed, helps to remove the irritating effect in the gastrointestinal tract by diluting or neutralizing the irritant substances. Sympathetic stimuli can also increase salivation a slight amount, much less than the parasympathetic stimulation. The sympathetic nerves originate from the superior cervical ganglia and travel along the surfaces of blood vessels wall of the salivary glands. A secondary factor that also affects salivatory secretion is the blood supply to the glands, because secretion always requires adequate nutrients from the blood. The parasympathetic nerve signals that induced copious salivation also moderately dilate the blood vessels. In addition, salivation directly dilates the blood vessels, thus providing increased salivatory gland nutrition as needed by the secreting cells. Part of this additional vasodilator effect is caused by kallikrein, secreted by activated salivary cells, which in turn act as an enzyme to split one of the blood proteins, an alpha-2 globulin, to form bradykinin, which is a strong vasodilator. To summarize the regulation of salivary secretion, salivary glands are controlled mainly by parasympathetic nervous signals from the superior and inferior salivatory nucleus in the brainstem. These nuclei are excited by both taste and tactile stimuli from tongue, mouth and pharynx. Especially the sour taste increases salivation 8 to 20 times. Presence of smooth object in mouth increases salivation in comparison with rough objects. Salivation also controlled by appetite area of the brain as well as taste and smell areas of cerebral cortex and amygdala. Reflexes originating in the stomach and the upper small intestine also increases salivation. Sympathetic stimulation can also increase salivation, but it is less than parasympathetic stimulation. Blood supply to the gland also affects salivary secretion. Parasympathetic stimulation causes vasodilation. Activated salivary cells also secrete kallikrein, which produces bradykinin, a potent vasodilator.